Hello everyone, my name is Yongsoo Kim. Today I will talk about the social brain and autism. We as humans, we are social beings. We like to be with friends and family. As you can see in this picture, children love to play together. And if you're left alone from a group of other people, you often feel sadness and lonely. Children with autism have troubles in engaging in normal social interactions. Autism is characterized by three distinct behavior impairments. The first one is impaired social interaction. They have a hard time engaging in normal social interaction. And the second one is they have communication problems. And lastly, they have repetitive behavior and restricted interest. So what happened in autism? How do we understand how autism happens in our body? To do that, we need to first understand our brain. Brain is a central information processing machinery in our body. And to understand how brain works, we need to understand basic unit of the brain, which is called brain cell neuron. And we have trillions of these. And these individual neurons talk to each other through a specific structure called synapse. At this synapse, each individual neuron has about tens of thousands of neurons. And so what makes this amazingly complex connectivity and this biological machinery? It's a gene. Gene contains information to build this amazingly complex machinery. So recent advances in the human genetic study identify several promising candidate genes in individuals with autism. And very interestingly, many of the autism candidate genes involved in generating proteins playing a critical role in synapse. To remind you, synapse is a communication portal for one neuron to communicate another neuron. So let's think about it this way. Brain is a chipset. Each chipset is composed of many different subunits. And these subunits need to talk to each other. When there's problems happening in this communication part, you may have problems. So is our brain. When neuron cannot communicate with the neighboring neuron, our whole brain information processing may fall apart, thus cannot generate the proper behavior output. So how do we study this? And one approach is using animal model. And it turns out that our favorite animal model, mouse, are also social animals. So we scientists can sit and watch how mouse socialize each other. So let me explain how it works actually. So we can take genes identified in the individual with autism, then put that particular genetic mutation into the mouse, and then see how mouse actually behave. So bottom is normal mouse. When mouse sees the other mouse engage in their version of social interaction, such as nose-to-nose touch and close following. And when a mouse has these particular genetic mutations, they tend to sit in the corner, do not engage in interacting with other mouse. So it's interesting, right? You take these genes found in human, put it in a mouse, and you make mouse less sociable. So now the next big challenge is, let's look at the brain. How does these particular genetic mutations change brain information processing to make this behavior output? So to do that, we develop a new microscopic techniques that can see each individual cells in the great detail in the whole mouse brain. So with help of the specially generated mouse, they can report brain activity. We can watch what brain area get activated when one mouse sees the other mouse. So these are the combination of brain areas that get activated when mouse engage in the social interaction. So we are currently using these techniques to understand how the mouse model autism process social information differently from this normal mouse. So what we want to achieve here is basically by understanding how animal model process social information in their brain, we can translate this information into the human brain and then get the insight how human brain process social information, thus understanding how autism arises in our brain. Thank you for your attention.